about stem cells, right? So what else we discussed? First we discussed the difference between stem cell and normal cell. What is the major difference? Tell me, students. Between stem cell and normal cell. It has an indefinite amount of cell divisions. It can undergo unlimited cell divisions, but whereas a normal cell will undergo a programmed cell death. That is called as programmed cell death is called as, I given a word for that. Yes, apoptosis. Apoptosis, right? Programmed cell death. Maybe after 30 generations or 60 generations, these cells will diminish. They can't replicate again, right? This is the normal property of a normal cell. Then next one, stem cells has three properties. What are the three properties of stem cells? Renewability, very good. Renewability, potency, plasticity, very good. So what do you mean by renewability? You know?
Produce more.
Unlimited cell divisions. Unlimited cell divisions without undergoing any differentiation. What is the meaning of differentiation? Conversion of stem cell to some any type of cell, right? Stem cell to skin cell, stem cell to columnar cell. Any cell can be, differentiated. That phenomenon is called as differentiation, right? Then potency. What is the meaning of potency?
The change in every cell.
Yeah, it is the ability of a cell to transform into any other type of cell. If it has high potency, that means it will be able to transform to wide variety of cells. So based on the number of cells it can transform, variety of cells when I say, they are of 3 or 4 types. What are the 4? If it can able to convert any type of cell in the body, entire body cells, that is called as totipotency. Totipotency. Right? Both totipotent cells are only present in the embryo. Totipotent cells are present only in the embryo. Right? Whereas next one is pluripotent. Pluripotent means it can undergo any 200 types of cells. It can undergo almost 200 types of variety of cells, right? Pluripotent cells. Then what is the next one? Multipotent. It has some limited capabilities, maybe around 20 to 60 cell types, right? That is called as multipotent cells. Then last and final one, unipotent. Unipotent stem cells, they will only undergo only one type of cell. Okay, this will be transformed to only one type of cell. Maybe that is one hair follicle, for example. Hair follicle has hair follicular blastocyte that will be converted to only hair and it cannot be converted to different cell. Okay, that is called as unipotent cell. So these are the various potencies of the stem cells: totipotent, pluripotent, multi, and totipotent. Unipotent. Then next one is plasticity. What is the meaning of plasticity? What is the meaning of plasticity means? The ability of a cell to the ability of a cell to convert back to the stem cell and to change to some other cell. For example, we have a cuboidal cell. Somehow cuboidal cell became a stem cell. Now this stem cell again converted to a columnar cell. Okay, the ability of the stem cell to convert to different form can be shown as a plasticity. So based on the environmental stress, it has the potential to change to its change its shape. That is called as plasticity. Just like a plastic material where we can mold it to any other shape, right? Plasticity. Then I want to discuss one more. Then I will discuss very interesting things here. Next one is I'm just writing. First we discuss about the definition, then we discuss the properties. Here we discuss the properties. Next one, types of stem cells. Stem cells themselves were divided into types. Okay, types of stem cells. Types of stem cells based on their origin. Okay, types of stem cells based on their origin. This is my cross statement, okay? It is a bracket. Based on their origin, stem cells are further divided into three categories. What are those categories? You have a sperm, ovum, you fertilized them on a petri dish, then you got a morula, okay, a underdeveloped baby, we can say zygote. From zygote, we know that it has a totipotent cells, harvesting a totipotent cells. Such type of cells are called as embryonic stem cells. These cells, harvesting of these cells is called as embryonic stem cells. Okay. Now, next one. We are taking stem cells from a full-grown adult. Such stem cells are called as adult stem cells. Harvesting cells from the adult. Okay, harvesting means corruption. Harvesting cells from the adult are called as adult stem cells. Is there any possibility that a full-grown adult like me having the stem cells in my body? What type of cells those are? Can you give an example? Bone marrow. Very good. So bone marrow. Yeah. So what type of stem cells present inside the bone marrow as per their potency? Yeah. As per their potency, what type of stem cells are in bone marrow? Multipotent, pluripotent and multipotent stem cells are present. Pluripotent because they can be converted to any type of blood cell. Multipotent if you take a single lineage. The cells that committed to become a red blood cell, the cell that committed to become a white blood cells, the cell that committed to become a lymphocyte and monocyte. Yeah. So this is it. That is called as adult stem cells. Bone marrow is one example. Next one, third one. Very, very interesting. Induced pluripotent stem cells. Induced pluripotent stem cells. Induced pluripotent stem cells. Okay? That I will discuss what is this. Very, very interesting. So now we know the types of stem cells based on their origin. If you are taken from embryo, embryonic, from adult, and this infused. I will discuss that, okay? Next important thing, before discussing all these stem cells, I just want to give you a grand picture. You know, this is really interesting. How many cells we have in our body? Trillions and trillions of cells present there. Just to take red blood cell, a drop of blood have 5 million of cells. That is a normal value, by the way. A single drop has 5 million red blood cells. That's why you are studying 4.5 to 5.5 million cells per cubic millimeter of blood. Per cubic millimeter means a single drop. Just imagine how many drops leads to 1 liter. That means how many cells present in 1 liter of blood. And we have around 5 liters of blood. Just RBC alone we have trillions. If we consider all body cells, you know, we have trillions of cells. You know, greater than the entire universe, stars in the universe. So we are very complicated beings, right? My question was, you just imagine all this data to make this complicated human being, either neural cell or anything, eye is anything, right? All this data stored in a single cell. And the data was split in 50-50%. 50% data present in the father, 50 data present in the mother. Right? Meiotic division, where the 46 chromosomes underwent, you know, partial division. What is that? 23 chromosomes. In the mothers, stored in the ovum, whereas in the father, stored in the sperms. When they connected again, we will get again 46 chromosomes, full grown, you know, full genes in a single cell. Right? So 23 in the sperm, 23 in the ovum, connected together to get 46 chromosomes. Together these 46 chromosomes in a single cell, keep in mind. In a single cell, 46 chromosomes have the information sufficient enough to make a full-grown baby. Just imagine how complicated that is. These cells or the chromosomes have huge amount of information. Huge amount of information. I'm telling again. Each and every cell carrying 46 chromosomes. That means theoretically each and every cell capable of becoming an entire human being if we allow it. Have you understood what I said? Because that is what exactly happened at sperms and ovum. These chromosomes splitted, they underwent substantial cell division, meiotic division, where the cells are splitted into half. Now, this is the same idea came to one person. What he did was, let me take 46 chromosomes, assuming these 46 chromosomes can able to make a full-grown human, having the data to make a full-grown human, right? Then, what is this concept of stem cells? Are stem cells the embryonic cells? Why stem cells? What I was trying to say was, listen very carefully. In this genome, in this entire genome, assume it converted to a type of cell, maybe a pluripotent hematopoietic stem cell. Now, in this pluripotent hematopoietic stem cell, there are this 100% human genes. Out of 100% human genes, only the blood cells require 3% of gene in one area. Maybe in the chromosome 24, we need only chromosome 24 should be activated and all other chromosomes must be inactivated. Okay? And that's what exactly happened in stem cells. In this committed cell, all other genes are inactivated except the genes responsible for only making blood cells, and all other genes are inactivated. Technically, those genes can be activated, but these genes have been suppressed. This is called as gene switches. The cell will turn on the switch, it will switch on the gene, or it will switch off the genes. In stem cells, there are various switches. That means various parts of the DNA got activated, and that DNA making the only that series of cells. You got my point? Now I am discussing at genetic level what are stem cells. Right? That means every cell has say 46 chromosomes, but not all 46 chromosomes working. Only few chromosomes will work as per the cell. Got it? Then how? A stem cell, how a mature differentiated cell is different from stem cell. In the mature differentiated cell, that cell genes are not ready to undergo any further evolution, further, change in the shape. They are only sustaining the cell metabolism, that's it. In the mature adult cell, in a normal skin cell, it is only surviving for its metabolism. Taking the nutrients, cracking of the 32 ATPs, it is only working at that level, but it's not working at genetic level to transform itself to some other thing. Right? That means meiosis not happening, sexual division not happening, or division that required to transform the cell. Have you understood what I said so far? Simple, keep in mind. In the 46 chromosomes, in one chromosome, maybe chromosome 40 has the data of making liver. So in the liver, there are a set of stem cells. They are only activating that 40th chromosome, but all other chromosomes are inactivated. That 40th chromosome can make any one of the liver cells. It can make a hepatocyte, it can make a gallbladder, it can make any portal system of the liver. Okay? So keep that in mind. Then I want to discuss induced pluripotent stem cell. Imagine, I have taken a liver cell, a hepatocyte. I know that in the hepatocyte I have 46 chromosomes, and I also know that only 40th chromosome number 40 was activated, and all other cells are deactivated. Other chromosomes are deactivated. Okay? Now, is it possible to take a hepatocyte and to reactivate the other genes, maybe chromosome 30, chromosome 20? Can I able to reactivate those other chromosomes? If I activate other chromosomes, definitely if chromosome 20 able to code a heart cell, cardiocyte, I can convert a hepatocyte to a cardiocyte by activating chromosome 20 in that same hepatocyte cell. Assume this is an adult cell now. Can I able to convert an adult cell back to something else, maybe a heart cell, by activating those genes? Is it possible? It's not. It might be possible. Maybe on theory it might be possible if you activate the genes. The genes you have activated and it should be converted to activated genes will start making proteins, appropriate proteins, and the proteins lead to that particular cell. Got my point? Then, who is controlling these molecular switches? Is our next question. We understood what are stem cells, but stem cells also need some gene activations. Since it is because of those gene activations, they are converting to blood cells, they are converting to neural cells, they are converting to other cells. Who are controlling those genes? What factors contributing to activate those genes? We don't know that. Got my point? What factors controlling the genes? Is it possible for a switch off gene to turn it on? I have taken 46 chromosome cell. Can I able to turn on the inactive genes? Is it possible? Then they understood this. They thought maybe all these 23 chromosomes will be activated. Maybe they are activating in some microenvironment. Maybe the environment facilitating them to get activated. Maybe we are assuming environment might be facilitating. These environments are called as niches. Niches, niches. Very interesting thing. Niches. Niches, okay? Niches, niche. Niches, niches. I will discuss niches after, and I want to discuss one interesting fact with you. Do you know how bone marrow transplant works? Even I was surprised when when I was first time heard by my faculty. You know how bone marrow transplant works? If you assume, what is your name?
Anishwar.
Anishwar. Anishwar, what is your name? Naman. Let us assume we are going to transplant Naman's bone marrow to the Anish. Okay? What you are expecting? Am I going to drill a hole in his bone? Am I going to drill a hole in his bone and I am going to drill her? I will take her bone marrow and drilling, injecting in his, that is called harvesting or transplanting into his bone marrow. That's not how the transplantation works. Very simple thing. I will give her some tablets. These tablets makes her bone marrow blood cells to come outside. They will come to her normal blood circulation. Then I will connect just her blood. That's it. I will do apheresis actually. I will connect her blood to a washing machine, a type of washing machine you can feel. Okay. That washing machine will spin, and I know that my stem cells has a particular size. They are large. Now, to my washing machine, have you seen inside the washing machine, there will be a small pouch. In that pouch, it will catch the dust inside the washing machine. So, I also kept a pouch in my washing machine. The pouch pore size is same size as the stem cell size. Okay, now the stem cells will be collected in the pouch. This machine is called as apheresis actually, apheresis machine. Now I have isolated her cells and I recirculate normal blood back into her body. So I am not wasting her complete blood. I just collected her cells. This is how I collected. Okay? Filgrastim is a tablet or injection I am going to give her, where that it will stimulate the blood cell, the stem cells to come outside. Then I did the apheresis and I have collected stem cells in a test tube. After collecting these stem cells, what I am going to do in the Anish after I am collecting from Ahan? This is very interesting. I will simply do a blood transfusion, that's it. I will keep a blood bag. I ask his blood group. If he said A group, A positive, I will take A positive blood group, patient of A positive blood. To this blood bag, I will introduce the blood stem cells. Now, something miraculous phenomenon happens. The stem cells, after going to Anish, They have a phenomenon called homing, H-O-M-I-N-G. Very, very, very interesting. Somehow, the stem cells present in his circulation will start migrating towards his bone marrow. No one knows how that migration happens. But stem cells knows where they belong in the body. If I inject neural stem cells, they will migrate to the neural system. If I inject bone marrow stem cells, they will migrate to their bone marrow. If I inject the cardiac cells, they will migrate to the heart. Somehow stem cell knows their home. How is this possible? How they are doing that journey in the body? You got my point? So this phenomenon is called as homing. One of my friends who met with an accident, whenever accident, he was parked in a junction, traffic signal. He has a very big bullet. So he got his leg like this, and there is one more student backside, classmate. He's waiting for the signal to turn green. Unfortunately, one car came and it hit, it went through the leg. And he got a fracture, major fracture here. Very painful, okay. So we all were sad, they took him to the nearby hospital. There is been 1 week, they kept rods. And the doctor shown us a X-ray report. And I was shocked, there's X-ray. You know what is the problem? Doctor haven't joined his both bones. Doctor kept some gap. And I asked him, why you didn't connect it properly? His surgery went properly. Then he said, that is how a surgery will be done. We are not going to completely enclose the bones. We will keep some gap. But the gap is major, not so small. Even I, non-radio technician, understood there's a gap in the X-ray. Then I asked him, so what's going on? And he said that gap will be filled by bone. Automatically it will be filled. But not skeletal system, not ligaments, not any other cells, but pure bone. When I say bone, not only bone, along with the bone marrow, everything is very complicated structure. Then I asked the doctor, how is it possible, sir? And you know what is his answer? Even I don't know, that works. I'm a surgeon, I will do surgery, but it will work, maybe God. Yeah. Then, there comes the stem cells. They know how to maintain their type, their territory. Got it? Now, the bone marrow has some chemicals, and these chemicals guiding the cells to become what? The niche, the environment. When there is a bone marrow environment, you keep whatever stem cell that will be converted to a bone marrow. In the cardiac environment, you keep whatever stem cell, it will be converted to the cardiac cell. The environment playing a vital role. Now you understood what is homing. When we given just cells, those cells are everywhere in the body. Those cells present everywhere, but the cells nearby the bone marrow started activated. Why they are activated? That activation facilitated by the bone marrow environment. That bone marrow niche or environment triggered the genes of nearby stem cells. What type of genes? To make the bone marrow cells, to make the bone marrow macro. So those cells started transforming to the nearby bone marrow. Such a wonderful concept. Okay, this is how transplantation works. Then, there is another interesting thing. The same thought came to one scientist. Okay, so now I understood I need to switch on the genes. If I switch on the genes, I can able to do whatever I want. I can convert the cell to whatever cell I need. Then what he did, maybe ovum and sperm, inside there is 23 chromosomes. This ovum has some chemicals, those chemicals activating all 23 chromosomes, so they are forming a baby. Maybe when we kept ovum and sperm together, they are making a hyper environment where they are activating all 46 chromosomes. This complete chromosomes activated means it might become a baby. Technically, maybe this cell, this ovum, sperm and ovum has the microenvironment to activate all the genes. Got my point? Then what they did? They did wonderful experiment. You listen to the experiment now. Now, I want a clone. The problem with the humans is this is a male and this is a female. Okay. See here. Here I have 23 chromosomes. Here I have 23 chromosomes. Now the baby will have half father genes and half mother genes. Half father genes and male and female. Alright. That's why we want to look like our father or mother. We look in between. Some father characteristics and some mother characteristics, right? Now, can I be able to make my clone? If I want to make same Vamsi-like person, my same 23 genes should be present in the ovum also, and that's not possible naturally. Am I correct? If I need a complete clone of a Vamsi, I want some 23 chromosomes in the ovum also, but I want a female. What should I do? So the scientists understood the game. What they did was, they took a ovum, they removed the 23 chromosomes from this ovum, they removed the chromosomes of the mother. Then they took the sperm 23 cells, okay? They injected these cells here. Actually, they haven't done this task, they done a very simple task. You know what they did? They just took the ovum, okay? Then they took the 46 chromosomes from my buccal cavity. Take my cheek, extract 46 chromosomes, keep in the ovum, just stimulate with the sperm. Now it started becoming a baby without a mother, without a second person. The 46 chromosomes got activated, and they, you know what happens once it grown? This became a sheep. They did this on a sheep, and that sheep is the first cloned animal. That is Dolly the sheep. This is how Dolly came into existence. Dolly is the perfect replica of its mother. In fact, it is not a single Dolly. Almost they did 7 Dollies. 7 sheeps came out of this experiment. Okay, cloning. Cloning can be done. The problem with cloning, already these chromosomes are old chromosomes, adult chromosomes. Chromosomes have something called centromere, 3 arm and Q arm. At the end of this, arms, there is a portion called telomere. The length of the telomere determines the age of the cell. Smaller kids will have large telomeres, but older people will have less telomeres. Literally, if we keep our telomeres alive, if we keep a healthy telomeres, we will be more teen. We will be more younger. So, but that's not possible. So, the problem is, since we are taking from the adult, already telomeres are less. Now we are expecting it to become a just born baby. Now this baby will have a quicker aging. Dollies died within a matter of months, but they died with more aging actually. In 7 months, they have shown the attributes of 40 years of age because their telomeres are already deteriorated. So, in one way or other way, embryonic stem cells or adult stem cells won't work out much for transplantation purpose. Okay. Is there any other way to do these experiments ethically without destroying the embryos? There comes the induced pluripotent stem cell that I will discuss in the next class. Very interesting. So one scientist came, he took the adult cell and he thought he made it reverse actually. He took a hair cell and he converted hair cell to a heart cell. And that scientist got a Nobel Prize. His name is Shinya Anamaka. He found a cytokine called OFX3. So there are 4 cytokine mixtures and he understood the game. So induced pluripotent stem cells has a plethora of applications that we will discuss in the next class. So far clear, right, about stem cells? Yeah.